This is the defense of the human body, smaller than the eye can see. Disease could be fatal to the body if these defenses weren't on hand to meet the challenge. And meet the challenge they do, constantly battling the hostile world of germs and bacteria with a more versatile and amazing army than you could ever imagine. Your body's main line of defense is made up of the lymphatic system and an assortment of white blood cells, each with a different job and some with several jobs. The lymphatic system is a network of millions of tiny vessels. Numerous little knot-like structures called nodes, which look like this under a microscope. And the fluid carried in the system, the lymph. The vessels are a lot like veins. They even have valves, ensuring a one-way flow of lymph away from the body tissues where it originates and into the major veins near the heart. There it enters the bloodstream. But exactly what is lymph? Why does it leave the body tissues in the first place? And what happens during its trip through the lymphatic vessels?
All the open spaces that you can see between the cells contain lymph. Lymph is nothing more than tissue fluid that has collected water and other molecules and sometimes foreign particles. This fluid constantly needs filtering to remove these various particles that it's picked up. Once it enters the special system of lymph vessels, that's exactly what happens. Along the way, it passes through several nodes. These nodes are filters with openings lined with special cells capable of removing harmful organisms and waste. Once cleaned, the lymph fluid is sent back to the main bloodstream for redistribution throughout the body. There are higher concentrations of nodes in different parts of the body. And while they're highly effective filtering devices, in some cases they can become swollen and painful. But your body has another means of defense against the hostile world of germs: white blood cells. And since they're the first to rush to the scene of the invasion, they're actually your first line of defense. There are 5 different types of white blood cells, and their names and the functions they perform can get a bit confusing. So let's look at them individually. Of the 5, there are 3 which have chemical granules in their cytoplasm. Each of these performs a specific function according to the type of granules it contains. This one has granules that stain red. These granules contain an enzyme that helps this particular cell to digest the foreign materials. Next, we have granules that stain deep purple. In this case, the granules are anti-clotting agents, and if the germs enter the body through a cut, these cells help keep the area from clotting, so more white blood cells can get to the scene and join in the battle. And finally, this cell has granules that stain light purple. They're usually the first white blood cells at the scene of invasion, by the way. They help engulf and digest bacteria. So we have 2 cells left that don't contain any granules at all. First, the monocyte, which is very large, and the lymphocyte, which is smaller. Now, of the 2, the lymphocyte is the most fascinating. It can actually change itself into other types of cells if the situation requires. For example, we call it a lymphocyte if it's working in the blood, but it can work outside the blood too. And when it's outside, it's called a phagocyte. In either case, the function is to remove particles.
These are the lymphocytes engulfing 2 colonies of bacteria in a drop of blood. You can see they don't waste any time. The white blood cells rush from all directions to attack and destroy the bacteria. In the bloodstream, they move freely among the red blood cells to seek out the enemy. Watch the bacterial colony on the right almost disappear. It's obvious in this case, at least, the bacteria don't stand much of a chance. We can get an idea of exactly how effective they are by injecting this rabbit with a solution high in bacteria. A few minutes later, if we take a blood sample for observation under a microscope, we can see bacteria are present, the black dots. But another sample taken after only 30 minutes shows a different story. There are many lymphocytes or white blood cells, but practically no bacteria. But we mentioned that white blood cells aren't restricted to the bloodstream. If the germ or bacteria happens to be outside the blood vessels, the white blood cells can actually squeeze through the capillary walls and take up the battle in the surrounding tissue. Here's how it looks inside your body. The white blood cells stick to the walls of the vessels. And then the cells actively squeeze through the walls of the vessels. Once in the tissue, the white blood cells move on, seemingly at random. The cells migrate into the inflamed area. But how do white blood cells destroy the enemy? To put it simply, they engulf and, in a sense, devour them.
Here they're engulfing the bacteria called streptococci. And here they're attacking rod-shaped bacteria called bacillus. Now the white blood cell begins digestion. White blood cells are one of the body's most important inner defenses. But sometimes the foreign material that we've talked about isn't bacteria or germs. In fact, often it's damaged or dead cells that were once a living part of your body. You're about to see a cell die. Watch closely, it happens pretty fast. There. Now watch the surrounding white blood cells. They start to devour it immediately. In case you missed that cell dying, watch again at the upper right. exactly how lymphocytes know when to attack another cell is something we're not really sure of. But with the use of a microscopic laser, we can explore one theory.
This red blood cell in the center is about to be destroyed. The white blood cells move in right away. Watch closely. Here's another. And the same response. They even seem to fight over the remains. We think this kind of reaction is caused by a chemical attraction, a kind of scent given off by dying cells. To illustrate, in this sequence there's a flow or current which might sweep the scent away. You can see there's very little reaction by the white blood cells. But let's take the flow away so that the scent remains. There's little question about how effective white blood cells are as combat soldiers in your body's army of defense. Close combat, though, isn't the lymphocytes' only talent. They also defend the body by helping to produce what we call antibodies. Antibodies are specific chemicals produced in response to an invasion of foreign particles. Put simply, they're a highly specialized type of soldier, produced to fight one particular type of invader. We can best illustrate how they work by thinking of them as being similar to pieces in a jigsaw puzzle. That is, fitting only specific shapes. When certain harmful agents enter the body, specific antibodies are produced. They, in a sense, fit the makeup of the invader perfectly and are then able to destroy them.
Often, the invaders are bacteria, which are comparatively large and easy targets for the antibodies. Sometimes, though, they're viruses, and that's a different story. For one thing, viruses are smaller than bacteria and much more elusive. Another complication is the way that they work. Instead of simply destroying a cell, they actually attach to your chromosomes and control your body cells for their own destructive purposes. The results simulated in this lab experiment are often things like abnormal chromosomes, or unusual cell divisions. This cell is about to divide. The one at the left has no nucleus. In another case, other mutant cells may be produced. How viruses are able to affect cells this way is almost like something from a science fiction movie.
We can use a model of the cell to illustrate. This virus attaches itself and injects its genetic material. Once inside, the genetic material behaves like part of the cell itself. The virus genes join with the cell's genes. Once this happens, they redirect the gene's reproduction, so it no longer reproduces cells like itself. Instead, it produces viruses. Finally, the cell bursts, and the new viruses are free to invade other cells. These are actual pictures taken with the help of an electron microscope. In this virus, the tail fiber attaches to the cell and pulls it close to the cell wall. Then the tail pins make contact. Now, a sheath around the tail contracts, exposing a slender central core. The core is forced down through the cell's wall like a hypodermic needle. Now, the viral DNA is actually injected into the cell, taking control. The added DNA has all the information needed to make new virus particles. Now, instead of producing cell protein, the cell is producing virus particles. The virus is using the cell to reproduce. In some cases, it only takes about 30 minutes for the cell to become filled with newly formed virus. Then they dissolve the cell wall and spill out. As more cells dissolve, more viruses are released. More cells are infected, and the infection spreads. So the next logical question is, how does the body go about fighting off something as small and destructive as a virus or any foreign invader? Well, one way is called phagocytosis, which simply means eating. We've seen too, though, that viruses aren't easy targets, so phagocytosis doesn't always work. A more effective means is something we've already talked about, antibodies. You may remember that we said antibodies are a specific kind of soldier produced to fight a specific invader. The shape of the antibody matches that of the invader. The way they're produced is another of your body's amazing functions. A few of the white blood cells that have come in contact with a certain virus have managed to escape carrying the invader's code. Some make the way to the lymph nodes, where they use those stolen codes to produce cells capable of making antibodies. Those are the plasma cells. In effect, the plasma cells start to build a highly specialized army of antibodies, now equipped to fight the battle. The army multiplies and is sent out into the body through blood vessels. This time, when they come in contact with the invaders, they're equipped to win. They work in several ways. This cell is attempting to engulf a harmful streptococcus, but it has no antibody to help it, so the streptococcus wins. You can see the cells slow down and eventually disintegrate. If we add antibodies, though, it turns out to be a different story. With the streptococcus inactivated, the cell has no trouble engulfing the intruder. Even as sophisticated and effective as the antibody system can be, in certain cases there's one fault with it. It takes too much time. Normally, the first time your body is invaded by a virus, it takes about 5 days for the antibodies to be released into the system. We call this first reaction your body's primary response. If you happen to encounter the same invader a second time, though, it's a different story. Some of the plasma cells containing the invader's code remain in the lymph nodes. So, when the next invasion takes place, instead of taking 5 days, the antibodies are released immediately to the scene. and the invaders don't stand a chance. This second type of reaction is called a secondary response. And because of that quick response, we say you become immune to that particular invader. Fast response or immunity is also the main factor behind another familiar word, vaccinations.
Vaccination was first proved valuable by Edward Jenner in 1796. He found that if a person were injected with the harmless form of a virus, the person would not contract the disease, but the antibodies would be produced. In other words, the primary response would take place. Then, in the future, if the vaccinated person came in contact with the virus again, the much more effective secondary response would take place. Antibodies would overwhelm the invaders before they could get a foothold in the body. That's immunity. In the case of some destructive invaders like smallpox, influenza, and polio, a primary response of 5 days might be too late. But suppose the invaders aren't viruses. Suppose they're very toxic poisons, like those in a snake bite. Obviously, it's impractical to vaccinate everyone against every kind of poison on Earth. So instead, when someone is bitten, they're given a specific antitoxin against that particular venom. An antitoxin is simply an antibody solution that has been made in advance by the bodies of other organisms, such as a horse. The horse is injected with a small amount of the poison, and its body then produces antibodies against it. These antibodies are contained in the blood serum, and they can be separated, bottled, and injected as needed into humans. There's no question your body's defense system is both an amazing and effective one. Like any other part of your body, though, there are times when it malfunctions, so it's just not effective enough. These are cancer cells in the process of division. Only one succeeds, however. We know that the mechanism of division is often defective when compared to normal cells. Here, a normal cell on the right, adjacent to a cancer cell, is about to divide. When the actual division occurs, it is marked by a violent motion. We don't know why. This cancer cell is dividing, but instead of the normal 2-cell division we would expect, 4 cells are produced. And 2 of these fail to even separate, leaving a single cell with 2 nuclei. This cancer cell, after separating from one cell, joins with another. And then separates again. Here, lung cancer cells display poor adhesion to one another. This poor adhesion may explain why some cancer cells spread. Your body's defense system isn't completely defenseless against cancer, though, as many people think.
With time-lapse photography, we can get a first-hand look at how lymphocytes attempt to destroy cancer cells. The tiny white cells with the very active motions are lymphocytes. The others are living cancer cells. Here you see a lymphocyte move within a growth of cancer cells. Inside this cell, an abnormal white vacuole forms on the left. This will result in the death of the cell. Here are 2 lymphocytes moving in cancer tissue. Do they somehow cause the explosive death of the cancer? We think so. In fact, in some cases, we've seen how it's done. You're about to see film of a cancer cell with a nucleus and 2 dark nucleoli. 2 lymphocytes are already inside this cell, and a third one is on the surface. Now this cell will eventually penetrate the cancer cell, slide past the nucleus, and join the other 2 white blood cells. We're viewing only this much of the cancer cell, and we're viewing it from above. So this cell right here is going to look as if it's on top of the nucleus. These are 2 lymphocytes which have actually broken into a much larger cancer cell near its nucleus. A third takes on an arrowhead shape, trying to penetrate also. It keeps trying. And eventually there's the breakthrough. Like many of the other functions of the body, we've still got a lot to learn about its defense system. There's one thing we do know, though. It's there 24 hours a day, fighting, keeping you alive.